Tanto biasa tujuan. Yang di sini ini pakai kayu.
Hi, Maggie.
Hi there. Hi, everybody.
Yes, that's good. I still managing the things to be done. 5 minutes left.
Yes,
Okay,
yeah.
we can wait. Mbak Tiara, Assalamualaikum. Sehat selalu. Mbak Emi, monitor.
Waalaikumsalam, Pak.
Sehat selalu ya. Oh,
Sudah
mantap.
terdengar.
Sudah kedengar ya. Mbak Emi, kopi Mbak Emi.
Iya, Bapak.
Weh, siap. Itu yang dari PAI sudah hadir kah?
Dari PIO Pak Agus ya?
Belum ya, belum ada ya?
Belum ada sepertinya, Pak.
Oh, belum ada. Yang mengganti Bu Yuli siapa nanti sambutannya?
Air bisa nggak sih?
Yang mengganti Bu Yuli itu ini siapa? Sambutan.
Pak Agus.
Bagus. Terus yang dari PI pusatnya?
Oh, kalau dari Badan Lambung nanti saya, Pak. Kalau
Oh
dari
ya siap.
pusatnya Pak Agus.
Bagusnya sudah masuk belum kah? Ya, Pak Agus, kita cari di mana? Pak Agus sudah kasih linknya ya?
Sudah, Pak.
Mungkin mohon dibantu dicek supaya Pak Agus bisa masuk ya. Ini belum ada di belakangnya. Bu Luna sudah kontak Pak Agus kah?
Sedang dihubungi, Pak.
Iya, iya.
Sudah dihubungi ya. Kalau sudah nanti bisa kita sudah mulai ini sudah jam 1 masalahnya. Itu kurang 1 menit.
Baik, Pak.
Sebagai informasi terdaftar 142 orang, ini baru hadir 30 orang ya. Sambil jalan nanti kita bisa mulai, tapi ini yang penting Pak Agusnya nih sebagai lagi we will wait some time for waiting for Pak Agus to join.
Would you like me to share my presentation?
Not yet, not yet.
Okay.
Will take. Bu Luna, apakah bisa mungkin dimulai sambil menunggu Bagus atau gimana ya?
Mohon maaf, tunggu sebentar, Bapak sedang
Ya,
dihubungi.
sebentar ya. Oke, terus kita.
Punten Pak, ini Pak Agus sudah jawab lagi proses buka laptop.
Oh, siap.
Ya, Pak.
Oke, kita tunggu. Posisi tambah 144 yang daftar, yang hadir 34. Nanti sambil selanjutnya pada berdatangan. Maggie will inform you that the participant will be registered about 130. Sorry. 145. But yes, still many people to come. So, dulunya dikabari kalau Bagus sudah join ya.
Baik, Pak. Sepertinya Pak Agus sudah gabung, Pak.
Sudah masuk?
Sudah.
Baik, kalau sudah masuk, kami serahkan ke Mbak Emi, langsung aja Mbak Emi.
Baik, Pak. Good afternoon, ladies and gentlemen. My name is Emi Sintadewi, and I will be your Master of Ceremony for today's webinar. On behalf of the organizing committee, I warmly welcome you to today's webinar entitled Western Beekeeping and Mite Control Strategies of Varroa SPP and Tropilelaps SPP, held on Thursday, March 5th, 2026. It is a pleasure to welcome our distinguished speaker, honored guests, colleagues, and participants this afternoon. Allow me to extend our respectful greetings to the Honorable Chair of the Indonesian Entomological Society, represented by Professor Dr. Agricultural Science Engineer Agus Tarno SPMP, the Honorable Secretary General of the Indonesian Beekeeping Association, Mr. Didik Purwanto, the Honorable Chair of the Indonesian Entomological Society Branch Bandar Lampung, represented by Ms. Luna Lukvitasari SSI MSI, our Honorable Moderator, Ms. Tiara Sayusti MSI, our distinguished speaker, Dr. Maggie Gill, and all participants who have joined us on this afternoon. We now proceed to the opening session. We will begin with the welcoming remarks from the Chair of the Indonesian Entomological Society, Branch Bandar Lampung, who is represented today by the Secretary, Ms. Luna Lufitasari, SSI, MSI. Ms. Luna, the floor is yours.
Okay, thank you, Miss Emmy. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Om Swastiastu, Namo Buddhaya, Salam Kebajikan, Rahayu. Honorable Chair of the Indonesian Entomological Society, represented by Prof. Dr. Agus Tarno. Honorable Secretary General of the Indonesian Beekeeping Association, Mr. Didik Purwanto. Honorable Moderator, Ms. Tiara Sayusti, MSI. Our distinguished speaker, Dr. Maggie Gill, and all participants who have taken the time to join this webinar. Let us express our gratitude to Almighty God for His blessing and grace, which allowed us to gather in good health and high spirits for the Road Toward Bee Day 2026 webinar, with the topic Western Beekeeping and Meat Control of Varroa SPP and Trophy Lelap SPP. Today's topic is highly relevant to the challenges of modern beekeeping. The development of Western beekeeping requires more intensive and science-based colony management. At the same time, the threat posed by parasitic meats such as Varroa SPP and Trophy Lelap SPP has become a critical issue that demands effective, sustainable, environmentally friendly control strategies. We would like to extend our sincere appreciation and gratitude to our speakers for generously sharing their knowledge and experience. We also thank all participants for their enthusiasm and active participation in this webinar. And this webinar is part of a series of events leading up to celebration of World Bee Day 2026, which will be held in Bandar Lampung on June 2026. On this occasion, we are also pleased to announce the activities and officially launch the World Bee Day 2026 website. The World Bee Day 2026 seminar will carry the themes Bees for Sustainable Palm Oil, Conservation, Biodiversity, and Productivity Enhancement, featuring distinguished speakers in their respective fields, including Professor Dr. Damayanti Bukhari, Mr. Dwi Asmono PhD, Mr. Hari Purwanto PhD, and Professor Dr. Hamim Sudarsono. Further information regarding the event can be found on the website. In closing, may this webinar bring broad benefits and serve as a strong first step toward the successful organization of World Bee Day 2026. Wassalamualaikum warahmatullahi wabarakatuh. Thank you.
Thank you, Ms. Luna Lukvitasari, for delivering the welcoming remarks on behalf of the chair. We also appreciate the information shared regarding the upcoming World Bee Day seminar on June 2026. Next, we invite the chair of the Indonesian Entomological Society Central Board, who will also officially open this event. Today, the central chair is represented by Professor Dr. Agricultural Science Engineer Agus Tarno, SPMP. Professor Agus, we respectfully invite you to deliver your remarks.
Okay, thank you so much. So sorry, I just got the notification a few minutes ago. So sorry, I can't prepare many things, but it's okay. I think it's not so important for me to provide the long speech, but the most important is the day this event is close to be the event that can be a part of the role of our Indonesian Entomological Society. So it must be related to our contribution to the peoples. So today we would like to thanks to, of course, Indonesian Entomological Society Association in Bandar Lampung, and also distinguished speakers. There are several entomologists there, and also I found also Professor Damayanti Bukhari, yeah. Also speaker from Dr. Maggie from Australia, maybe, and also several members from Indonesian Entomological Association. Of course, it's not only in Bandar Lampung, but also several area in Indonesia, and also all colleagues that also join with our events. So today we have it is great pleasure for us to join with this webinar in the celebration of the World Bee Day, an important moment for us to recognize the extraordinary role of bees and other pollinators in supporting biodiversity, agriculture, and food security. Actually, we are not only celebrating bees, but we are also highlighting the important contribution of Indonesian Entomological Association in promoting knowledge, research, public awareness about insect and their role in our ecosystems. Indonesian Entomological Association actually has played a key role in advancing entomological research, supporting scientific collaboration and sharing knowledge with the farmer, student, policymaker, wider community, and etc. However, hopefully this meeting also can be one of the media that can support such kind of increasing the skills and also increasing the information for all of us, and also related to the publication, outreach activities, and many things related to the science and also society. Before I close this speech, actually, I want to inform that our president cannot join with us because of several agenda. So I should replace him to be one of the person that can that can join with these events. And also, I do hope that the next is not only in the World Bee Day. Hopefully, we are the community of entomology in Indonesia can also join and make a partnership together. It's not only with international scientists, but also many communities in Indonesia. And we are waiting for that. And of course, we also want to increase the collaboration between international community. Another thing that also important is the problem that already happened in case of bees. Yeah. So today we will also get information about how we can control the problem that can reduce the productivity of the bees. So hopefully many people can get much more information related with that. And finally, I would like to express my appreciation to Indonesian Entomological Association, and also especially in Indonesian Entomological Association in Bandar Lampung, and all partners who continue and contribute to scientific development and environmental conservation. And let us continue working together to ensure that pollinator, including bees, remain a vital part of our ecosystem and our future. And that's all, and thank you. And hopefully this webinar can provide many new insight and also many information, and also many things related with how we can increase the productivity of our bees. Thank you so much. I have give back the time to the moderator. Thank you.
Thank you, Professor Agus, for the remarks and officially opening this webinar. Ladies and gentlemen, we now move to the main session of today's webinar. It is my pleasure to hand over the session to our moderator, Ms. Tiara Sayusti, MSI. She is a doctoral candidate at IPB University and a biology lecturer at Universitas Pertahanan Indonesia. Ms. Tiara, the time is yours.
Okay, thank you very much, Miss Emmy. Okay, Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. First of all, maybe I would like to thank first the organizers of the World Bee Day 2026 from Indonesian Association Apiaries in collaboration with Entomology Association in Indonesia, branch Lampung, especially Lampung Province, right? For inviting me to be the moderator for the second webinar in this World Bee Day series. And as introduced by Miss Emmy, my name is Tiara Sayusti. I'm a doctoral candidate at the Department of Biology, Faculty of Mathematics and Natural Sciences, IPB University, and a biology lecturer at Indonesia Defense University. And now I'm very happy to share that today's participants come from very diverse backgrounds, including beekeepers and also lecturers, researchers, and also students from various universities across Indonesia. And before we begin, here are a few quick reminders. Maybe first, please mute your microphone during the presentation session. And we will have a Q&A session after the presentation. And please kindly write your question in the chat box, or you can ask directly to the Dr. Maggie to ask the question after the presentation. And finally, it is my great pleasure to introduce Dr. Maggie Gill from United Kingdom. Maybe later we will call you Ibu Maggie. Is it all right?
That's good. Yes.
Yeah, to be more feel Indonesian. Yeah, maybe let me first read a short CV of Ibu Maggie first. So Ibu Maggie is the founder and principal researcher of Vira Science, Pollinator Health International Research Association, a non-profit organization dedicated to improving the health of pollinators globally through the robust scientific research. And she is also an experienced commercial beekeeper with 20 years expertise in producing honey, colonies, queen, and she is now specializing in research-driven approaches to pollinator health. And for her education, she got the Bachelor of Science honors at the Natural Science major at Open University, UK, and MSc of Entomology at Harper's Adam University. And also she is currently pursuing her PhD in the chemistry major at Keele University, UK. And for your information, she is currently visiting Indonesia to meet Professor Dr. Ricardo Feudin, right, from Department Biology, IPB University, as her research counterpart. As her research counterpart to plan a collaborative study on the Tropilailaps mite affecting honeybees in Indonesia. So during her visit, we invite her to share her fascinating experience and also practical insight in working with honeybee ectoparasitic mites in this webinar. And we are truly honored to have you with us today, Ibu Maggie. Thank you very much for being with us. So without further delay, I would like to invite Ibu Maggie to start her presentation entitled Western Beekeeping and Mite Control of Varroa spp and Tropilailaps spp. The time is yours.
Thank you so much for that kind introduction, and thank you everybody for having me. It's a great pleasure to be in Indonesia and to meet beekeepers and to be able to talk to you all. So I will just share my screen, hopefully. Can you all see my presentation now?
Is it on progress?
Oh, sorry. Yes, one moment. Okay. Can you see my screen?
Not yet. How about the other participant? It's worked now. It works now.
Okay. Sorry. I think the internet is a little bit slow, but hopefully everybody can see that. And I will just minimize this one out of the way. And so, like I said, thank you everybody for inviting me to talk to you today. I'm here to talk about Western beekeeping and the control of parasitic mites in sorry, one second, that we use in Western beekeeping. So as you know, I come from the UK. I've been a commercial beekeeper for about 20 years. For the last 10 years, I've worked for the UK government, and I worked for the National Bee Unit. So the National Bee Unit is the government department that is responsible for honeybee health in the UK. And they hire government inspectors, and I was one of those inspectors, to go out and to work with beekeepers in the United Kingdom to inspect their colonies for diseases and for pests. And this picture here actually shows me as a bee inspector, if I can just get the pointer. So this is me working with a commercial beekeeper to inspect their colonies for disease. Now, beekeeping in the West is quite different to Asian beekeeping. You can see that we have quite big colonies. So we have a brood box here where the queen is kept, and she is kept in this box with a queen excluder. And then above that box, we will have what we call supers or honey boxes where the queen can't reach and she can't lay eggs and produce brood. And this is where we collect our honey crop. So first, a bit about myself and Fire Science. So Fire Science was established in 2023 to carry out pollinator research, and we've focused a lot on honeybee health in the last 3 years. In particular, we've focused on the parasitic mites Varroa and Tropicalaps. So in Fire Science, there is myself, who is the founder, and I have 5 colleagues who also work with us, and we are all bee inspectors with the National Bee Unit. We have also all been commercial beekeepers with varying levels of experience from 15 years up to my colleague Kevin, who has 50 years commercial beekeeping experience. Recently, we've also started working with other government departments from around the world. So on the left-hand side here, we have Jeff Wilson. So Jeff works for the Canadian government. He is also a honeybee health inspector in Canada. He is the Saskatchewan provincial apiarist. He is a commercial beekeeper himself. And he does very much the same job that I do, where he works with beekeepers in Canada to monitor their colonies for health and to inspect for diseases and pests. And then on the right here, we have Irakli Janeshii. He, again, is a beekeeper of over 30 years experience. He works with the Georgian government, and he carries out a very similar role there, where he inspects honeybee colonies for health. And there are various different organisations that we have worked with over the last few years along the bottom of the screen there. So first of all, as I said, we come from many different countries in the West. So I thought I would first give you an idea of beekeeping in those countries, and I would start with Canada. So this first picture here, obviously Canada is very, very cold, and it has very, very cold winters. This first picture here was sent to me by my colleague Jeff Wilson about a week ago. And this is one of his apiaries. So there are, under this pile of snow, there are approximately 25 to 30 colonies. And Jeff keeps his honeybee colonies outside all winter. He wraps them in insulation, and then there is this black tar paper. And you can see that they go through the winter very well, like this. This is a colony that Jeff opened up in the middle of winter, and you can see that it's a strong colony and there's lots of bees there. To control Varroa mites, so to control Varroa mites, Jeff will use oxalic acid sublimation, and this is his skidoo, where he's gone out to one of his out apiaries. It has a generator on the back of it, and this is the oxalic acid sublimator, which is a long metal tool that you can insert into the entrance of the beehives. The electricity from the generator heats that up. You place oxalic acid on that tool, and when it heats up, it releases a vapor into the hive to treat Varroa mites. So not all beekeepers overwinter their honeybee colonies outside in Canada. Some beekeepers will overwinter in bee sheds. So this is one of those bee sheds, and they are climatically controlled In this shed, there are approximately 3,000 colonies. The colonies will be placed in the sheds in sort of late August, early September, and they will be taken outside again when the winter is over at the end of April and beginning of May. And they maintain the temperature at 4 degrees Celsius, and they maintain the humidity at about 60% humidity, and the bees go through the winter in these sheds. When the bees come out of these sheds in the winter, Canadian beekeeping, commercial beekeeping is very big business. So most Canadian beekeepers will have between 5,000 and 15,000 colonies. They don't migrate their bees. There are very large plantations of various different crops. So in this picture, you can see canola, which is a brassica plant that produces an oil. And their honey season starts in mid-May and runs to the end of July. So they have a very short 8 to 10 week season where the bees are actually working and collecting honey. So these two videos show some of that, the equipment that they use to extract this honey. So in Canada, there are approximately 800,000 beehives with 15,000 beekeepers. In 1 week, one colony can produce 35 kilograms of honey, and this factory, in 8 hours, it will process 20,000 kilograms of honey. Now, most of this honey is then placed into drums like this, and it is exported to predominantly Japan. It's a very valuable commodity, and it's very high quality. The Japanese markets will pay a lot of money for this honey. So then we move on to Georgia. So Georgia is a country that is on the border of Asia and Europe. So it's in Western Europe and on the border of Asia. You can see that they also have cold winters with snow, but they also have very hot summers. So this is a short video from the summer, and their climate in the summer is very similar to yours. So temperatures will range from 30 to 45 degrees Celsius, and they will have very high humidity of between sort of 70 up to 100%. So very similar to the summer conditions that you have here all year round. Beekeeping in Georgia is mostly migratory, so beekeepers will keep their colonies in the valleys during the winter, and they overwinter their colonies in the valleys. Most beekeepers have between 100 and 800 colonies, so smaller scale than Canada. So the Georgia is formed by the Caucasus Mountains, so they have two very large mountain ranges, and the migratory beekeepers will move their bees up these mountain ranges as the flowers come into bloom, and the bees will collect honey from things like acacia, sweet chestnut, and linden. And by August, the colonies will have reached the high alpine meadows, where they will forage on meadow flowers. That is their final honey crop. Once the beekeepers have collected that, they will move their colonies back down to the lowlands, where they will overwinter. And then finally, beekeeping in the United Kingdom. So this is where I come from. Beekeeping in the United Kingdom is mostly a hobby, and it is a pastime that people do for fun and for pleasure. There are very few commercial beekeepers in the UK. I believe there's only about 400 commercial beekeepers. Most beekeepers in the UK only have between 3 and 5 colonies, and they will keep them at their home in their garden. And generally, the honey that these beekeepers produce, they will sell it locally or give it to their friends. The minority of commercial beekeepers, so there is some migratory beekeeping where they will access crops like borage. So this is a borage crop, and there are also heather moors, which are up in the highlands of the UK. So beekeepers in the United Kingdom can register with the government. They don't have to, it's not compulsory. It's thought that there's about 100,000 beekeepers in the UK. As I said, approximately 400 of those are commercial beekeepers. But on average, beekeepers only have about 5 colonies. Our season is very short, so it runs from April to October. And if the weather is good, we can sometimes get 2 crops from that honey harvest. The average amount of honey that a colony will produce each year is about 15 kilograms. But you can see from this map of the UK that there are lots of beekeepers in the United Kingdom, and there is a very dense population of beekeepers and hives. So as I said at the start, I work for the UK government for the National Bee Unit, and as a National Bee Unit inspector, we will go out and we will monitor honeybee colonies for pests and for diseases. So we have 2 statutory notifiable diseases in the United Kingdom that we monitor for. That's American foul brood, so Penobacillus larvae, and European foul brood, Melissococcus plutonius. Now, the honeybee health in the United Kingdom is very well funded. When I am a bee inspector and I go out and I work with beekeepers, that is a free service, so the beekeepers do not need to pay for that service. If we find disease, those colonies are destroyed, but the beekeepers are paid compensation. And because of that, generally the beekeepers are very cooperative with the UK government, so we have very low incidence of disease in the United Kingdom. In 2025, we inspected 35,000 colonies. Only 63 of those had American foul brood. When we inspected for European foul brood, only 814 colonies had European foul brood. And we do not use antibiotics in the United Kingdom. We tend to destroy the colonies and pay the beekeepers compensation for those colonies that we've destroyed. We also monitor for 2 statutory notifiable pests, and we carry out inspections for those pests. Those pests are small hive beetle, Athena tumida, and Tropalaelaps. So we do not have either of these pests in the United Kingdom at the moment, but we proactively monitor for them. The idea is that if we can detect them at an early stage, then we will be able to eradicate them. Small hive beetle is currently in Italy, in Europe, which is somewhat close to the UK, and Tropalaelaps is in Eastern Europe. And we do import queens and bees from these regions, so there is a risk that we can import these exotic pests. Feral colonies, so swarms that have attached to lorries and shipping containers, they also pose a transportation risk. The National Bee Unit also do other work. So we have had a recent incursion of non-native Asian hornets, so Vespa valentina. They were first detected in 2016, but now, unfortunately, last year it's thought that Vespa valentina are now established in the southeast of the United Kingdom. But the National Bee Unit will go and find these hornets' nests and destroy them. The other work that we do, so we will randomly test honey samples to see if they're contaminated with things like miticides, antibiotics or pesticides. We will investigate honeybee poisoning incidents. And as I said, there is this insurance system in place for beekeepers. So if they lose colonies, they can place an insurance claim and they are compensated for their honeybee colony losses. And we act as the assessors for those claims. So the work that Fire Science does, so we predominantly focus on Tropalaelaps, but in the past we have also done work on nectar nutrition. So this table comes from a piece of work from 2018 where we looked at the different floral resources in the United Kingdom, and we sampled nectar from those plants, and we looked at the nutritional values that those nectars gave to different pollinators in the UK. When it comes to honeybee disease, such as American foul brood and European foul brood, we're also working on a project to monitor honeybee colonies as a way to detect subclinical levels of these disease so that beekeepers can preemptively treat for those diseases. And this project, we place samplers in the hive and then we test those samplers to see if we can detect changes in the volatile organic compounds that are indicative of disease. But the biggest piece of work that we've done for the last 3 years is working with Tropalaelaps. So we have developed monitoring techniques which the UK, European and Canadian and North American governments now use to monitor for Tropalaelaps. We've also done studies into how tropicolaps can be transmitted. So can they be transmitted in things like queen imports and packaged bee imports? We've also monitored their transmission in swarms. But I think the most important thing, and the thing that most beekeepers are interested in, is how can you control tropicolaps and varroa in your hives. Varroa has spread out of Southeast Asia. I believe it was the late 1950s where it was first detected in Apis mellifera colonies. And since the 1950s, every decade, varroa has slowly spread around the world. It reached North America in the 1980s. And then finally in 2022, it reached Australia, which was one of the final countries and continents where varroa has not spread. And to this day, varroa causes significant honeybee colony losses in the West. So in the United Kingdom, UK beekeepers will lose between a third to 40% of their honeybee colonies to varroa each year. The number is higher in the United States. So over the winter of 2024 and into 2025, beekeepers lost approximately 60% of their colonies to varroa. And now recently in Australia, since varroa has arrived there, beekeepers are experiencing high levels of colony losses. So varroa is probably one of the biggest issues that world beekeeping faces. Tropicolaps is an emerging threat. So Tropicolaps is native to Southeast Asia, and it naturally infests those giant honeybee species. But over the last few years, it has slowly spread out of Southeast Asia. It was detected for the first time in Europe in 2024, and this report that I have here also discusses how an Apis dorsata, so a giant Asian honeybee colony, reached the United States, and this colony was infested with tropicolaps mites. It was destroyed, so they didn't manage to become established in the United States, but it shows how tropicolaps is also spreading around the world, and particularly where it has reached Eastern Europe, it is causing significant colony losses in Eastern Europe and Russia. How can we control tropicolaps and varroa in our colonies? So as a beekeeper, you can use one of two different techniques. So you can use biotechnical control methods where you manipulate the colony in a way to create a broodless period so that the tropicolaps and the varroa cannot reproduce, or you can use chemical treatments. So in this video, my colleague Dan is using an oxalic acid sublimator to treat colonies for tropicolaps and varroa. So biotechnical controls, there are a range of different biotechnical controls that can be used. So for example, drone brood removal is quite effective for controlling varroa mites. Varroa will preferentially infest the drone brood. And as a beekeeper, you can cause your colony to produce drone brood and then remove this before the bees and the mites emerge. It is approximately 50% effective. Queen caging is another method of varroa and tropicolaps control. Queen caging has become particularly popular in the country of Georgia to control tropicolaps as well. And this is where you will go into your colony, find your queen, place her in a specially designed cage, and you will leave her in this cage for sort of 25 to 28 days. So the colony becomes broodless, and that break in the brood means that the tropicolaps and the varroa mites are unable to reproduce. Another technique is what we call in the UK is a shook swarm. And this is where you take all of your brood frames, you shake the bees from those brood frames into a new hive, and you give them foundation to work on, and you remove all of that brood from the colony. It is very effective. It's more than 95% effective. However, it does take a long time for that colony to recover because you're removing all of the brood from that colony. And finally, comb trapping is the final technique. This is where a frame of brood with the queen on is placed in a cage. It stops the queen from laying on other brood frames, and when that brood is ready to emerge, it is removed and destroyed, and therefore you're removing the mites that are present within that brood. Chemical control is also another popular option for controlling parasitic mites in honeybee colonies. Here I've put some pictures of different treatments that come from around the world, and you might be familiar with some of them. the most important thing to remember about these chemical treatments is that the majority of those have been developed to control varroa mites, and they've not been developed to control Tropicolaps. Varroa and Tropicolaps have very different life cycles. When a varroa mite enters the cell, they will produce a series of offspring, but when that bee emerges from that cell and the varroa mites emerge, those varroa mites need to be able to feed on adult bees in order to be able to reproduce. So varroa mites will spend between 5 and 7 days on the adult bees feeding. And it's in that window of time when the varroa mites are on the bees that the majority of these chemical treatments will work, because the active ingredients come into contact with the mites. Tropicolaps, however, have a much shorter phoretic phase, so they do not need to feed on adult bees in order to be able to reproduce. When they've reproduced within a cell and that honeybee emerges, they will emerge with that bee, they will go to the next available brood cell and they will start their reproduction, which means that they spend the majority of their time sealed within the brood, and the active ingredients in the majority of these chemicals don't actually come into contact with the mites and are less effective. So the exception to that is formic acid. So these are some formic acid treatments that are available around the world. Formic acid is an organic acid, so this has the added benefit that it's very unlikely that mites will be able to develop a resistance to an organic acid. It also has a very small molecule, which means that formic acid can penetrate into the brood cells, and it can actually kill the varroa and Tropicolaps mites that are reproducing within the brood. So we wanted to look at formic acid as a way to control Tropicolaps and varroa simultaneously in colonies. We looked at a couple of commercially available treatments that are available. So the first is Formic Pro. So this is a treatment that is licensed in North America and Europe. It is a 45% formic acid solution, which is in a gel matrix. You can see the pads there that you place on the top bars of your brood box. The second treatment that we looked at is Muravinka. Muravinka is a Russian treatment. It uses 85% formic acid, and there is sort of gel crystals, and you can see that they are contained within this small cloth bag. And again, you place this on the top bars of the brood box. And we wanted to see if these commercially available treatments, how effective they were at controlling both Tropicolaps and varroa in colonies. And we did this in Georgia, where Tropicolaps arrived in 2024. We did this experiment last year in 2025. So what did we find out? Well, you can see from these pictures here that when we did this experiment, that the temperatures were pretty similar to what you experience in Indonesia and Southeast Asia. So when we started the experiment, we put the treatments on at night. It was 27 degrees Celsius. Most of the time during the day, it was around 30, 32 degrees Celsius. And you can see that the relative humidity was actually quite high. So we've got 94% humidity there, 72% there, and 85% there. So we put the treatments on our treatment colonies, and at the start of the experiment, we monitored the bees. So we assessed how healthy were the colonies, how many frames of bees did those colonies have at the start of the experiment, were all of the queens healthy? We assessed how much brood were in those colonies. So this picture here shows a frame of brood, and for every colony we counted, so these are 5 centimeter squares, we counted how many squares of open brood there were and how many squares of sealed brood there were to give us a baseline of how healthy the colonies were. And then to assess the levels of mite infestation, we sampled sections of these brood, and we opened up the cells and we counted the number of live and dead Tropicolaps and varroa mites. This took a very long time. We spent quite a lot of late evenings doing this microscopy work. So what did we find out? Well, the first trial we ran was in August, and you can see that the temperatures were relatively high, so about 30 degrees Celsius with high relative humidity. And for the Formic Pro, so we checked the colonies after 60 hours, so after about 2.5 days, and we found that Formic Pro killed 97% of the Tropicolaps mites after 60 hours, and over 99% of Tropicolaps mites had been killed after 5 days. Muravinka wasn't quite so effective, so after the 5 days, this had killed approximately 88% of the Tropicolaps mites. One of the benefits was that we didn't notice any negative effects on the colonies, so we didn't lose any queens, we didn't see any dead bees or dead brood. However, this treatment wasn't very effective for Varroa. You can see from the results here that even after 5 days, both treatments had not killed a lot of Varroa mites. So the Formic Pro had only killed approximately 43% of the Varroa mites. Muravinca was not as good, so had only killed about 22% of the Varroa. So in terms of research, this was quite good because we'd managed to kill the Tropalaelaps mites, which had been one of our goals. But as a beekeeper, these results weren't very useful for us because this meant that even though we had killed the Tropalaelaps mites in the colonies, we still then had a high Varroa infestation, and we would need to go on and to treat those colonies for Varroa and use a second treatment, which is not ideal as a beekeeper. So we decided to run the experiment again. So we went back in October, several months later, and we did the experiment again. So everything was exactly the same. We used the same treatments. And you can see that we had very different results when we ran the experiment for a second time in October. So you can see from this graph, after 60 hours, the Formic Pro and the Muravinca, so both treatments had killed over 99% of the Tropalaelaps mites. So very, very effective at killing Tropalaelaps. And from this graph, so this graph shows the Varroa, after 60 hours, over 95% of the Varroa mites had been killed. So we had very good efficacy with both treatments. But the drawback to this was that we did lose some queens. So some of our queens did die. And you can see from this picture that the treatments did kill quite a lot of the adult bees. So we had piles of dead bees outside one of the colonies. And the big difference between August and October was the ambient temperature and the ambient humidity. So the main thing was actually the humidity. We had very high humidity in August, so over 80%, but in October, the humidity actually dropped to between sort of 50%. And we think that it's the ambient humidity that is affecting the way that the formic acid is released into the hive and penetrating into the cells. So we didn't stop there. We've actually worked on some other delivery methods. We were pleased that we've managed to kill a very high percentage of Tropalaelaps and Varroa mites, but obviously it's not ideal to kill queens or to kill the bees. So we've actually tried some of these different forms of delivery methods for formic acid. So this is a sheet of blotting paper, so it's an absorbent paper that we've tested. We've got various different pads here. And this is a form of sponge that we've used. So we've had some very good results from this, and we're hoping that we can spend 2026 developing these different delivery methods and techniques to be able to deliver formic acid to colonies where we have very good efficacy and we kill a lot of the Tropalaelaps and Varroa mites, but we're not harming the colonies. Another thing that's important for beekeepers is being educated about Tropalaelaps and Varroa. This is a page from the Phytoscience website. On that website, we have lots of free resources for beekeepers. So there are educational videos for beekeepers on there on how to detect Tropalaelaps, things about their life cycle, the symptoms of Tropalaelaps and Varroa that you will see in a colony, and also how to treat your colonies. And also, if you're interested in producing your own presentations, there are videos on there which are free to use. So if you want to go and give a presentation to other associations within Indonesia, please feel free to download these videos and use them yourselves. There's also all of the research that we publish is on the website and can be accessed there. And we post things on social media as well, which will keep you up to date with the latest research and our latest findings with treatments for Tropalaelaps and Varroa. Thank you for listening to my talk, and I'd be very pleased to take your questions.
Okay, thank you very much, Ibu Maggie, for the excellent and insightful presentation. It is very amazing what you work, I think, and very, very helpful for us, yeah. So now we will move to the Q&A session. Anyone would like to ask a question directly? Please feel free to unmute your microphone. or you can give a reaction of raise hand. So I will notice you. Sorry. Let me check the room chat first. There is one question. Actually, Pak Hasbullah and Pak Nazri have the same question. Is there any biological way to control the bee mites, or especially plant-based biological control for the varroa or tropilila mites?
So in the West, so in the United Kingdom and Europe and North America, we will quite regularly use thymol. So that is an oil that comes from oregano. So that's a natural
oregano.
Yeah, so that's a natural plant oil that can be used. It's very effective for varroa control, but one of the issues that you have with using thymol is that it will only target the mites that are outside of the brood cells. So in the UK, we have quite strict regulations about when you can use treatments on colonies. You can't use treatments when you're collecting honey. So we have very short windows of time when we can use these treatments. But if you use something like thymol and you're able to leave them on the colonies for a longer period of time, as those bees emerge and they come out of the cells, the tropilelaps mites and the varroa mites will come out, and the thymol does actually affect them. It stops them from feeding. So it is a very natural way to control mites in the hive. I'd also recommend formic acid and oxalic acid. They are both organic acids. The mites cannot develop a resistance to them. And I think particularly formic acid, because it has that ability to penetrate into the brood cells, that's very effective at killing both mites. And you can use quite short treatments to control them, so that you're not having to use a chemical on the hive for a long period of time.
Okay, thank you very much. Maybe I will translate first your question to the questioners. Okay, Pak Hasbullah and Pak Nazri, ya tadi dikatakan oleh Bu Maggie, ternyata ada ya plant-based control untuk varroa atau tropilila mites itu menggunakan minyak yang diproduksi dari oregano. Nah, oregano itu salah satu tumbuhan, mungkin nanti bisa di-searching ya. Oregano ini biasanya bisa dipakai juga untuk bumbu masakan ya. Jadi bisa di-searching oil dari oregano ini dapat digunakan untuk mengkontrol varroa atau tropilelaps. Nah, namun tadi juga sempat disampaikan bahwa formic acid itu efektif ya, lebih efektif untuk mengkontrol atau membunuh ya dari mite varroa atau tropilelaps ini, karena dia mudah penetrasi ke dalam larva sehingga bisa cepat gitu membunuh varroa atau tropilelaps tadi. Begitu. Apakah cukup Pak Hasbullah dan Pak Nazri?
Cukup, terima kasih.
Okay, thank you very much. Thank you very much. Okay, maybe there are several questions from the chat box. Okay, the second question comes from Pak Hari Purwanto. Hello, Pak Hari. Pak Hari is one of Ibu Rika College from Gajah Mada University at Yogyakarta. Pak Hari, I will read the question. Could the tropilelaps feed on adult honeybee?
So until very recently, it was not thought that tropilelaps could feed on adult bees. However, you have to remember the biology of their natural hosts, Apis dorsata. So Apis dorsata are a migratory bee species, and very little is known about how tropilelaps and Apis dorsata coexist and co-evolve. So Apis dorsata will have periods of time where they have no brood. and it's not known how Tropicolaps survive, but potentially they are able to survive on the adult bees. So I did a piece of work in 2024 where we had Tropicolaps mites survive for up to 5 days on adult bees, Apis mellifera bees, and up to 9 days on dead Apis mellifera bees and larvae. So they can feed on other things apart from live honeybee brood, but there's not much known, and I think they probably can feed in some way because of the way that they've evolved with Apis dorsata, but we're not really sure of that mechanism at the moment.
So Tropicolaps cannot feed on the adult bees, right?
Well, it's not known, but they can survive. So they can only. It's thought that they can brood, but they can survive in broodless conditions.
Yeah.
People don't know how they survive, so it's
Yeah,
a
okay.
answer.
Okay. If we compare to the larva cannot survive if infected by a Tropicolaps.
Yes, yeah.
Then adult bees. Okay.
Yeah.
Thank you very much. Okay. Ini we have two participants that will ask directly to Ibu Maggie. Please, maybe Pak Afri. Pak Afri, silakan Pak Afri.
Oke.
Mungkin memperkenalkan diri dulu ya Pak.
Okay,
Yeah.
thank you. I do apologize because I cannot open my camera. It's a very bad signal right here. So let me introduce myself. My name is Afri. Now I'm working for Ministry of Forestry in Pekanbaru. So yeah, it was a very nice presentation, open our perspective about how we deal with Tropicolaps. And I have at least 2 questions here. The first one is what is the most significant devastation caused by the Tropicolaps? Because in Varroa we already know that the Varroa can make deforming virus, right? So it's very devastating for the honeybee. And is it similar to Varroa? And I mean, the Tropicolaps brings the virus also. And which one is between those mites leads to the most devastation? Because Tropicolaps in Indonesia is less studied compared to Varroa. And my second question is, what is the latest study status about the honeybee vaccine? Because in the 2 or 3 in the back, we already knew that the vaccine already tested in several honeybee colony, and it shows that a good result. And what do you think about this result? Is this a prospective and some kind of a breakthrough of the how we manage about the Varroa? Okay, thank you. I think that's all of my question.
Thank you for your question. So first, the differences between Tropicolaps and Varroa. So Tropicolaps and Varroa are very similar because they breed and they reproduce in honeybee brood, and they feed on that brood in order to be able to reproduce. The main difference with Tropicolaps is the way that Tropicolaps feed is actually more damaging than Varroa. So Varroa might have piercing mouth parts, and they make a very neat wound when they feed on the honeybee larvae. It doesn't cause too much damage to that pupae. Tropicolaps have tearing mouth parts, and when they tear the pupae. If it doesn't kill the pupae, those wounds will go on to develop into deformities in the adult bee. So you will see much higher levels of deformity. So not only deformed wings, but deformities to things like the proboscis and the antennae. So the damage that Tropicolaps cause when they feed is actually higher than that of Varroa mites. Both Tropicolaps and Varroa vector viruses. The difference with Tropicolaps is they vector slightly different strains of virus to those that Varroa introduce. Now, in the West, that's important to us because our bees being naive to Tropicolaps. They have no resistance to these new viruses that are introduced. So particularly in Georgia and Russia, where Tropicolaps have arrived in the last sort of 2 to 3 years, we're seeing very high colony losses. So over 80% of colony losses are dying. And it's thought to be a combination of the way that the Tropicolaps feed and these new viruses that are causing issues for the bees. And then I think another concerning issue with Tropicolaps when you compare it to Varroa is that it has a faster reproductive cycle, and it's much more mobile. So it spreads from colony to colony much more easily, and then when it has spread, it reproduces more quickly, so it produces more offspring. So they are very similar, but there are these differences which make it difficult to control Tropicolaps, and particularly in the West where it's a new pest, very big issues with it.
How about the answer, Pak Afri? Do you want to give a feedback?
I think the second question, what is your opinion about the honeybee vaccine?
So I think the honeybee vaccines are very good, and they're a very positive step towards helping protect our honeybees from these viruses. I think one of the issues is that it's quite hard to deliver the vaccine to the colonies, and also the cost and the time that takes to deliver the vaccine. So hopefully with more work, the cost will come down and that delivery method of the vaccine will become easier, because I think for it to be successful, we need to be able to administer the vaccines to a large population of honeybees. But the actual work and the research is very, very good. And I think it offers hope because things like deformed wing virus, they do cause colony losses and they do impact the honeybee health.
All right, very good. Thank you.
Thank you.
Thank you very much, Pak Afri, for the question, and thank you, Bu Maggie, for the answers. It's very clear, right? And if we looking back to the room chat, there are so many questions for you. Maybe I will choose the interesting one from Pak Ali Bosar. His first question: do Varroa and Tropicolaps attacks occur simultaneously or at different times?
So you will find Tropicolaps and Varroa in the brood cells at the same time.
Okay.
So, and certainly, so I've worked in Thailand. This is my first visit to Indonesia, but in Thailand, in tropical beekeeping, it seems that both mites are present in the colonies all year round because you have that continuous beekeeping season. Less is known about it in the West, where we have temperate climates and we have those harsh winters. Initially, we thought that the Tropicolaps wouldn't be able to survive the winters, but we've just had, so I showed you that picture of the snow in Georgia with the colonies in the snow. Tropicolaps might have survived that winter, and they are now sort of reproducing in the spring. So we have had Tropicolaps 12 months continuously in colonies in Georgia now. So that is worrying. So unfortunately, we do have both mites in the colonies continuously all year round at the same time.
Okay. Pak Ali, apakah ada feedback Bapak? Jadi tadi Ibu Maggie menjelaskan bahwa ternyata bisa ya Tropicolaps and Varroa bisa menyerang dalam waktu yang bersamaan gitu ya. Namun ada perbedaan nih kalau misalnya di iklim temperate seperti di Eropa itu kan ada winter ya, ada musim dingin gitu. Nah di musim dingin itu ternyata Tropicolaps itu dan Varroa itu tidak bisa survive. Jadi berbeda dengan di Indonesia yang merupakan negara tropis sehingga perkembangannya itu continuous karena tidak ada musim dingin. Jadi mereka bisa survive sepanjang tahun begitu. Kira-kira. Silakan disampaikan feedbacknya mungkin kalau tidak langsung tidak apa-apa via chat. And anyone would like to ask Ibu Maggie directly again? If there is no question, this is one question from the chat room from Bapak Tulus ya. Ini in East Kalimantan. Let me read the question. In East Kalimantan, where brood is continuous year-round, humidity is high, and most beekeepers are small-scale with additional environment pressure from plantation and mining. Is it realistically possible to maintain stable Apis mellifera colonies without routine acaricide use against varroa destructor or Tropilailaps? If so, what practical biological indicators and infestation threshold should be used in the field, not in the laboratories, to prevent colony collapse?
So I think, unfortunately, I think Apis mellifera, they have no natural defenses to Tropilailaps or varroas, so beekeepers will need to continuously treat their colonies for these mites. I've worked sort of through Asia and into Europe, and the answer is always the same. If beekeepers do not treat their colonies, they will very quickly succumb and collapse from mite infestation. So I think, unfortunately, it's not realistic to maintain stable colonies without a routine treatment. I think what beekeepers can do is learn to monitor and to monitor the levels of mites in those colonies and treat, rather than just continually treating, just treat when it's appropriate and when the mite thresholds, I would say anything sort of above 10 or 15% of Tropilailaps or varroa infestation, you need to start thinking about treating those colonies. If you go to the Phytoscience website, there are various videos on there on how beekeepers can monitor for Tropilailaps and varroa. These techniques, so we've developed them in the field. You can see when we video them, it's us in an apiary doing these techniques. And if beekeepers proactively monitor and then treat appropriately as needed, I think that's a bit more sustainable than some of the situations that we currently have.
Oke, thank you very much Bu Maggie. Jadi jawabannya secara singkat harus di ini ya Pak, harus di monitoring secara rutin ya untuk perawatannya untuk supaya tidak ada Tropilailaps begitu. Jadi tetap harus dicek secara rutin, dimonitoring secara rutin apakah, dan kita juga harus bisa mengecek bagaimana level investasi dari si Tropilailaps ini. Dan untuk caranya atau metodenya itu bisa mengunjungi website dari Virascience yang tadi Ibu Maggie sampaikan. Di website itu terdapat beberapa metode yang bisa digunakan untuk melihat ya, mendeteksi bagaimana sih kita menentukan level investasi dari varroa berdasarkan metode yang ada di website. Jadi bisa dikunjungi website Virascience mungkin untuk mengecek ya video-video yang ada di website tersebut, Bapak. Begitu kira-kira, Pak. So, before I continue the question from the room chat, maybe anyone would like to ask directly to Ibu Maggie? Please raise your hand if you want to ask directly to Ibu Maggie. And Ibu Maggie, actually, when the participants registered in the G form, we also provide the column for the question. Let me check the question from the Google first. Wait. There is one question, interesting question from Pak Rustana ya. Why do newly emerged bees sometimes have damaged or deformed wings? Is it caused by varroa or what?
So I think it's difficult to tell, but if you have Tropilailaps and varroa in your colonies, so it can either be a virus, so deformed wing virus, which can be transmitted by Tropicolaps or Varroa. But as I said, when Tropicolaps feed, they can damage the brood, and that will cause deformities as well. So it can be a combination of both. But I think if you're seeing deformed wings, then perhaps you need to be thinking about treating your colonies and
Oke. Jadi Pak Rusnata yang tadi bertanya ya, ini di chat juga ada. Jadi sayap lebahnya rusak itu kenapa? Karena salah satunya dari virus ya, virus yang dibawa oleh Varroa atau Tropicolaps. Jadi kalau misalnya ada menemukan lebah yang dia baru menetas gitu ya, kemudian sayapnya kecil, itu kemungkinan dari virus yang dibawa oleh Varroa atau Tropicolaps yang menyebabkan sayapnya itu tidak berkembang gitu. Jadi perantaranya adalah sebenarnya penyebabnya virus, tapi perantaranya itu adalah Varroa atau Tropicolaps. Begitu, Pak. Okay. We still have lots of time. All participants, please, if you want to ask directly to Ibu Maggie, we will place. Pak Hari mau bertanya Bapak. Punten masih di mute Bapak ininya. Mikrofonnya Pak Hari.
Okay, thank you Mbak Sayusti. Nice to have you, Miss Gill. Actually, my undergraduate thesis was on Varroa and Tropicolaps mites in a year 1990. So it's long ago.
Pak.
Yeah, but we still have a problem with this mite here in Indonesia. And I think I already read some of your paper.
Publication.
Yeah, publication that my question is, we have more than Tropicolaps and Varroa destructor here in Indonesia. We have Jacobsoni, we have EU Varroa, we have Underwoody, and etc. So my concern is, do you have any suggestion that we should monitoring the possibility that the other mites other than Tropicolaps and destructor could also infest our mellifera beekeeping here in Indonesia? Thank you.
Yes, I think other mites are a concern. So, for example, U. Varroa, there's not been much research into U. Varroa, but there was, so in Western Australia in 2023, a colony of Apis florea was found at a port on a shipping container, and the colony was infested with U. Varroa. So it was removed and it was destroyed. But since then, so since 2023, a further, I think 43 Apis florea colonies have been found in that area, and they've all been infested with U. Varroa. Now, none of the research has found U. Varroa mites on Apis mellifera in the area, but since the Apis florea and the U. Varroa have arrived in that area, the Apis mellifera bees now have new viruses which they didn't previously have before this incursion of Apis florea. So there is evidence to suggest that these different mite species, while they might not be necessarily reproducing to a great extent in the Apis mellifera brood, they do seem to be transmitting viruses. And I think with anything sort of honeybee, so the mite research for honeybees, particularly Tropicolaps and Uvaroa, is quite limited, and there are many questions that we just don't know the answer to. But situations such as the one in Australia is concerning, because it does seem to suggest that Uvaroa have been able to infest Apis mellifera and have transmitted new strains of viruses to these bees. And I think just unfortunately just more work is needed to investigate this, because there's lots of questions that we don't know the answer to.
Thank you for the answer.
Thank you very much, Pak Hari. Thank you, Ibu Maggie. And we will back to the room chat. After maybe from Pak Budiman Harahap ya Pak, from Riau, from the Riau province. How do you monitor the strength of your queen bees after the winter season? Is there any research on improving queen genetics? From which countries or regions do the best queen lines originate for queen genetic enhancement?
So I think there's lots of research into improving queen genetics, and certainly Apis mellifera, there are lots of subspecies of Apis mellifera. I believe you mostly have Apis mellifera ligustica here, which is an Italian, or originally came from Italy, potentially via Australia and then into Indonesia. I think what I've found working with bees over the past 20 years is that the best Apis mellifera that you can have is one that's locally adapted to your area. And as a beekeeper, I spent quite a lot of time with my own stocks, just selecting my best stocks. So whether that was for honey production or docility or Varroa resistance, and just selectively breeding from there, because if you use your own local stocks, they will naturally adapt to the local environment. They will adapt to the different nectar flows and the temperature and the humidity. I don't necessarily think that importing different subspecies of Apis mellifera is the best idea, because they won't have that natural adaptation. And I think at an association level, in the UK, in Canada, and in Georgia, we have local queen breeding programs where beekeepers will get together and they will share their best stock. So I think the best bees that you can have are your local bees, but as beekeepers, you need to proactively select from your best ones and decide, I want lots of honey, I want them to be Varroa resistant, I want them to be Tropicolaps resistant, and then breed from your best colonies and keep very good records of which colonies are good and which colonies are bad, and be quite strict with what you select.
Okay, thank you. Thank you very much, Dr. Maggie. And maybe this one question is related to prior question from Pak Kuntadi. Do you think Varroa resistance queen can be effective to Tropicolaps infestation?
Sorry, could you repeat that? Sorry.
Do you think Varroa resistance queen can be effective to Tropicolaps infestation?
So I think we've looked at this partially in Georgia, and I think there are bees that have certain they will express certain behaviors that will control Varroa, and they will express certain behaviors that will control Tropicolaps, and if you selectively breed for those. So a lot of progress has been made with producing bees that tolerate Varroa and their resistance. Well, we've not had Tropicolaps in the West for a very long time, so we've not been able to develop those traits. But I do believe that with selective breeding, then certainly that would be possible. The traits might not be the same, so you need to, when you do your assessment and when you're breeding your queens, you need to look at both mites almost separately and breed from bees that actively control both mites rather than just one or the other.
Okay, thank you very much. And the one question comes from Pak Nurfianto from East Java, Malang, ya. So the question is, considering that formic acid is also found inside of bees naturally, what is the profile of formic acid content in stingless bees or honeybees and their defense mechanisms against mites? If there is no formic acid in honeybees, is it due to the breeding and domestication of honeybees?
So I'm not actually certain how much formic acid is actually present in honeybees. I know certain honeys have actually sort of quite high levels of formic acid, but I think whatever if there is formic acid present in the bees, it's not necessarily enough for that to be able to control so what you need is with the formic acid treatments that we use. So in the UK, we will use a dribble method of formic acid to control varroa mites. And that dribble method, because the mites are outside of the brood for between 5 and 7 days, when you dribble that formic acid into the hives, it comes into contact with the varroa mites and kills them. To control tropicolaps, though, because those tropicolaps might spend the majority of their time within the sealed brood, that's why we've used this sublimator to make that formic acid gas, because we want that gas to be able to penetrate into the brood cells. But I think, so while formic acid is effective at controlling both mites, I just don't think it's present in high enough levels in the actual bees to be able to control the mites, unfortunately. But it does work as an effective treatment.
Oke, thank you very much. Jadi memang benar ya Pak, ada di stingless bees dan honey bees itu memiliki formic acid juga, tetapi levelnya itu tidak cukup ya untuk bisa mengatasi investasi dari varroa atau tropicolaps tadi, sehingga memang membutuhkan kontrol dari bahan kimia dari luar begitu. Oke, maybe okay, all the participants say thank you very much on the chat box for Bu Maggie.
I thank you everybody for attending this talk. It's been a great honor to present my work to you and to talk to you about Western beekeeping and how to control these mites. So thank you for listening to me.
Okay, maybe one question from me. Last night I read your publication entitled Assessment of the Efficacy of Field and Laboratory Method for the Detection of Tropicolaps in 2024, I think. Maybe you can describe to us, especially to the beekeepers, because lots of beekeepers here join the webinar, maybe especially the method in the field for the detection of tropicolaps.
Yes, so this piece of work, we did the work in 2023, we published in 2024. So that piece of work was initially it was for government inspectors to use in the field as part of their inspection to monitor for tropicolaps and varroa, and we found that so the most effective technique for finding tropicolaps is to look in the brood. So if you take a pair of forceps and you have a frame of sealed brood, we would uncap 100 brood cells, pull out the pupae, and then just visually examine that brood for tropicolaps mites. And if you have tropicolaps present, if you uncap a minimum of 100 cells, if it's in the colony, you should be able to find it, and you should be able to work out a level of infestation from that. So 10% of the brood is infested, 20% of the brood is infested. I think if you're reaching levels above probably sort of 15% mite infestation, then you want to be thinking about treating your colonies for infestation. But we also developed some other techniques. So uncapping the brood by hand, it is quite labor intensive. You do have to sit there for quite a long time. If you look on the Phytoscience website, so there are those videos. So we've developed a technique called rapid brood decapping, which uses wax strips. So it's a sticky wax strip. You stick it onto the surface of the sealed brood, pull it off, and that decaps that section of brood very quickly. And then you'll see the tropicolaps mites will run out of the brood. So that's quite a quick and simple method to use. There's a video of that on the website. The other thing that we do is what we call a powdered sugar roll of the bees. So we take
sugar roll.
Yeah, we take a scoop of bees, We put them in a jar. We add a scoop of very fine sugar. So it's the sort of thing that you'd use to ice a cake with. Roll the bees in that sugar, so all of the sugar sticks to the bees. They then groom themselves. If you shake that sugar onto a white tray, the mites can't hold onto the bees, and the bees groom themselves. So you will see the mites in that sugar. So that's again, it's quite a quick way where you can just quickly check lots of bees and see if they have mites on them. But there are lots of techniques that beekeepers can use, but those would be the three that I would recommend as just quick and simple and reliable to find Tropicalaps and varroa mites in your colonies.
Yeah, thank you very much. Jadi, Bapak Ibu, let me translate your answer first. Dari papernya Ibu Maggie itu ada beberapa metode ya yang dapat kita gunakan untuk mendeteksi secara cepat bagaimana apakah ada investasi Tropicalaps atau varroa yang pada koloni lebah kita gitu. Beberapa metode itu ada di papernya, di publikasinya. Mungkin nanti saya akan coba kirimkan juga ya di room chat publikasinya Bu Maggie. Jadi pertama ada cara pertama itu yang agak lama itu uncapping atau membuka tutup ya, membuka tutup dari brood ya. Kemudian dicek gitu satu persatu apakah ada Tropicalaps-nya atau ada varroanya di situ. Nah, cuman itu cara itu tuh agak lama sih dan membutuhkan waktu dan mungkin agak bahaya juga ya bagi lebahnya, bagi larva lebahnya. Kemudian yang kedua ada menggunakan metode comb bump. Nah, comb bump itu dengan cara di shake gitu ya sisir sarangnya untuk membuat si varroa atau Tropicalaps-nya itu jatuh gitu. Dan kita melakukan itu di atas tray warna putih, white tray, untuk melihat apakah setelah kita guncang sedikit dengan lembut gitu, apakah ada varroa atau Tropicalaps yang turun. Nah, kemudian cara yang ketiga adalah dengan sugar roll. Nah, yang sugar roll ini adalah dengan cara lebahnya itu kita ambil gitu ya, kita gulingkan di gula yang halus gitu. Nah, si gula ini tuh fungsinya ternyata untuk melepaskan mites atau varroa atau Tropicalaps-nya tuh dari tubuh lebah gitu, sehingga dia mudah untuk terdeteksi jika kita gunakan gula tadi gitu. Nah, itu beberapa cara yang dapat kita gunakan untuk mendeteksi secara cepat apakah koloni lebah kita itu terinvestasi oleh Tropicalaps atau varroa. Demikian mungkin Bapak Ibu, mungkin one last question from, maybe I just see the question from Pak Hamim. Wait. Pak Hamim, I will read the question from Pak Hamim. Are there many people in England who truly make a living from beekeeping? Does it provide a good income in a year? Which months are usually the peak honey production season?
So yes, we do have commercial beekeepers in the UK. It's not the easiest job in the world, so our weather can be very rainy, it can be very cold, but there are many beekeepers that do manage to be commercial beekeepers. They tend not to just do honey production. They will also do things like produce queens and produce other colonies for sale. They will sell things like pollen, royal jelly, bee venom. So we have quite a diverse range of products that beekeepers produce in order to be able to make a living from it, and they will also teach beekeeping and charge people for lessons. So yes, you can make a living from it, but you have to be quite diverse in what you do and how you use those honeybee colonies to make a living from it.
Okay, Pak Hamim, do you want to give feedback? To Ibu Maggie. Nah, Pak Hamim open kamera. Thank you, Pak Hamim. Yeah. Okay. The last question from Ibu Awari Susanti. I will read the question from Ibu Awari. To what extent is the infestation level of Varroa and Tropilelaps mite parasites in bee colony, and is there a difference in the level of infestation between local and introduced bee species?
So in different regions of the world, the levels of infestation seem to vary quite differently. So currently in Georgia, where Tropilelaps is new, levels of Tropilelaps infestation are very high. But I think that's because the bees are naive to Tropilelaps. When you look at countries such as South Korea, where Tropilelaps and Varroa have been present for, I think, sort of since the 1970s, 1980s, the beekeepers find that Varroa levels of infestation are much higher than levels of Tropilelaps. So it's quite varied, and I think that's potentially due to the different subspecies of Tropilelaps Mercedes-i and Varroa that are found in different regions. When it comes to the different bee species, so Apis cerana and Apis florea, so these bees have naturally evolved with parasites, whereas Apis mellifera has not naturally evolved with parasites. So bees like Apis cerana and Apis florea, they tend to tolerate parasitic mites. So whether that's Varroa or Tropilelaps or Varroa, much better. So they tend to have just very low levels of mite infestation because they have those natural evolutionary strategies in place to deal with it.
Oke, so Ibu Erwani tadi ternyata berbeda ya. Ternyata apabila Varroa dan Tropilelapsnya itu menyerang misalnya Apis cerana atau Apis florea, itu ternyata Apis florea atau Apis cerana itu bakalan bisa lebih survive dibandingkan dengan Apis mellifera. Salah satunya itu karena untuk Apis cerana dan Apis florea itu bisa lebih mendeteksi ya, lebih mendeteksi dari investasinya itu, dan dia bisa abskon gitu, mencari tempat lain, membuat sarang lain yang dia bisa menghindari serangan Varroa lebih lanjut gitu. Oke, ini Bu Maggie juga sudah mengirimkan link ya, link untuk website dari Vira Science yang bisa diakses oleh Bapak Ibu apabila ingin melihat lebih lanjut konten-konten dari Vira Science. Banyak sekali, saya juga sudah mengecek tadi malam ya, banyak sekali konten-konten video-video yang sangat bermanfaat untuk bisa kita pelajari. Salah satunya tadi tentang deteksi dini untuk investasi Varroa dan Tropilelaps, dan masih banyak lagi. Ada siklus hidup dari Tropilelaps, Varroa, dan itu semuanya ditampilkan dengan sangat mudah dipahami. Jadi bisa dicek website dari www.virascience.org. Okay, yeah, thank you.
I've also put in the link for the National Bee Unit, so that's the UK government honeybee health website. Lots of free information on there. So I'd recommend on Varroa, on Tropilelaps, on all sorts of honeybee health things.
Okay, thank you very much, Ibu Maggie. So, all right, everyone, because there is no question again from other participants, and due to the time limitation, we will conclude the session. So today we have learned about Western beekeeping practical strategies to control Varroa and Tropilelaps mites, which are among major challenges in honeybee colonies also in Indonesia. And we hope this knowledge will support better bee health management and promote sustainable beekeeping in Indonesia. Thank you very much, Ibu Maggie, for sharing your valuable knowledge and experience with us today. Your insight on honeybee health and mite management have been very informative, especially for all of us. And we also thank all participants, beekeepers, lecturers, and researchers, and also students for your active participation. And with that, I will hand the session back to the MC. Thank you very much.
Thank you.
Okay, thank you, Ms. Tiara, for leading the session so excellently. Our sincere appreciation goes to our distinguished speaker, Dr. Maggie Gill from Parascience, United Kingdom, for the insightful presentation on Western beekeeping and effective mite control strategies. Before we conclude, we will now proceed with the presentation of a certificate of appreciation to our distinguished speaker as a token of our sincere gratitude. The organizing committee, please present the certificate. Okay, the certificate is proudly presented to Dr. Maggie Gill in recognition of her valuable contribution and outstanding presentation. Thank you once again, Dr. Maggie.
Oh, thank you so much for. I've really enjoyed this talk, and thank you for the certificate. It's very gratefully received.
Okay. Okay, we have now reached the end of today's webinar. On behalf of the organizing committee, I would like to express our deepest gratitude to our speaker, moderator, honored guests, and all participants for your active participation and enthusiasm. We hope this webinar has provided valuable knowledge and practical insights that can be applied in beekeeping practices and mite management strategies. Thank you for your attention and participation. Have a pleasant afternoon, stay healthy, and we look forward to seeing you at our next event.
Mungkin foto bareng dulu, Mbak Emi, boleh sambil dibuka kamera semuanya. We will do the photo session, Bu Maggie, with open camera all the participants.
That sounds good.
Okay. Maybe let me lead the photo session. Mbak Emi izin? Yeah. Okay. First slide. 1, 2, 3. Cheers. Sorry. The bubble chat is emerged. Okay. The first slide is done, and then second slide. Okay. 1, 2, 3. Okay. And then the last slide. The third slide. Yeah. 1, 2, 3. All right. Thank you very much, everyone.
Thank you.
Thank you, Bu Maggie. Thank you, Bu Luna, Bu Tiara, dan semua kawan-kawan. Terima kasih. Kita tutup ya.